So welcome, everybody. I'm so excited that you're all here. We're going to be hearing today from Dr. Simon Vanderkar, who, if you do not know, is an assistant professor of biostatistics in the Department of Biostatistics at Vanderbilt University Medical Center. And he's going to be talking to us today about effect sizes and replicability in brain-wide association studies, or BWAS. And I'm very excited. So leaving it over to you.
Thanks, Julia. Thanks for organizing this. Yeah, thanks for inviting me to speak too. So this is, I haven't given this talk before. This is work by one of my students, Kaidi Kang. It's based on, it sort of builds on other stuff we've been doing and some sort of discussion that's occurred in the literature regarding effect sizes. If there are questions, feel free to stop me throughout, because I don't think it's going to be that long of a talk. There's a lot of information in here, though. So there's two papers that came out recently. One of these is called was in Nature last year, and what this paper showed. Well, so let me tell you what a BWAS is. So brain-wide association studies are sort of a class of studies that are used to identify brain behavior associations, or maybe brain and age associations. Pretty much any time you want to understand the link between brain and some kind of phenotype, these sort of fall into the category of brain-wide association studies. And this recent paper by Merrick et al. That was published in Nature showed that correlations in brain-wide association studies are smaller than expected based on the sort of body of published literature. So the way they looked at this is they used the ABCD data set, which includes about 10,000 kids between, I think they're around 10 years old. And these kids have been scanned with MRI, and then they also look at a bunch of psychometric measurements as well, and include things like sort of IQ and measures of psychopathology. And what they showed in this data set, and then they replicated it in two others, is that the correlations are all very small, smaller than expected based on, like, if you look at smaller individual studies, the correlations are usually larger. And what they showed is, like, most of them are less than 0.1. And so, their whole argument was that, well, part of their argument is that, data mining, publication bias, and high variability of estimates in small studies leads to seemingly larger effect sizes. And so, like, if you look at this plot at the bottom, they show sort of the variance of the correlation coefficient over the sample size. And you can see, like, the variance is larger in smaller studies, and then it gets smaller and smaller and smaller, and it converges to, I forget what variable this is for, but you can see it's converging to some very small correlation value that's the observed value in this data set. So this is a bootstrap analysis. So they're saying basically, like, the variability that you get in sample sizes of like 50 or 100, and then that combined with people sort of looking for associations leads to publications that have a larger correlations than are realistic in the data set. And part of their conclusion about this is that this is a concern for replicability. And this is because what they suggest is that you need very large sample sizes for replicability. So this is another plot from their paper. This is the Merrick paper. And they showed that basically you need sample sizes on the order of 1,000 to begin to have reasonable statistical power. And that if you look at how, depending on how they define replication, but if they look at replication is less than power, and it's like the probability of replication is even still quite low. even with sample sizes, well over 1,000. So sort of my opinion about this is that, if this is true, that effect sizes are small in brain-wide association studies, it sort of uh, poses an uncertain future for the utility of MRI as a marker for psychopathology. If you have very small effect sizes, you have low discrimination between a patient and control population, which would mean that, it's just, if you need thousands of samples to determine an effect, that effect is probably not going to have much clinical utility. They did show, and some responses. The Spisak et al. Was a response to the Merrick et al. And they showed that multivariate BWAS have larger effect sizes. And so by multivariate BWAS, I mean neuroimaging studies where you're using the imaging variable as the predictor, and you're predicting the phenotype of interest. And so that's what they showed here. And sort of, there's a lot of information in this slide, but sort of, if you look at this first panel, these are the sample sizes that they required to get 80% power. And so they showed that, using different types of machine learning models. So, by machine learning models, I mean support vector regression or ridge regression, that your sample size can be in the hundreds and you still can have 80% power, however they define power or replicability in this particular situation. So larger effect sizes are expected when you're sort of aggregating information across the image. And sort of ad hoc, people have said they expect effect sizes to be larger in studies that are looking longitudinally. So basically looking at repeated measurements on participants. So, they were focusing on replicability, and, part of the question is what factors affect replicability in brain-wide association studies. So I found this definition of replicability from the National Academy of Sciences. Replicability is obtaining consistent results across studies aimed at answering the same scientific question, each of which has obtained its own data. So when you read this definition, it's clear that replicability doesn't mean anything unless you define what consistent means. And they don't do that. So, it's really dependent on how you define consistent. And, that's subject to, particular, to particular person or a particular field. But as replicability is often defined in terms of hypothesis testing, so, two studies having the same significant result, then it's easier to understand what factors affect replicability. And those things are the study population. So are you looking at, for example, adolescents or older adults? The bias of the study procedures. So, are people, is there an a priori hypothesis, or are people sort of like mining the data for a particular effect? And then it's going to depend on these two things sort of analytically, the sample size, and the effect size. Both of these are related to power, and replicability is basically a function of power, controlling for these two other things. So the Merrick study essentially said, sample size is something we can control, and, to have replicable studies, we need very large sample sizes. And so what I'm going to argue is that effect sizes are also something we can control, and that we can design studies in such a way to maximize effect sizes. Okay, so effect sizes are, let me I'm gonna minimum. Oh, I wanted to hide this, whatever. So effect sizes are that they're talking about in brain-wide association studies are standardized parameters. And one example of that is correlation coefficient. So it's sort of normalizes out any scale of the outcome in covariate distributions. Okay, so if you look at the correlation coefficient in simple linear regression, it looks like this. So you have your whatever the true parameter is in your population, and then you have sigma x and sigma y. So sigma x is the variance of your covariate, sigma y is the the standard deviation of your covariate, sigma y is the standard deviation of your outcome. And it's easy to think of these as unmodifiable parameters, but these things are actually modifiable parameters. So you can adjust the features of the study population by designing the study differently to increase this correlation coefficient. So, an example is you can increase the standard deviation of the covariate, and that will, deterministically increase this effect size. So what that might look like when you're collecting data for a study or designing a study is this. So if, let's say I'm studying age, and then I have some image outcome, such as gray matter volume, then in the first plot, this is the way people usually collect data, which is just if I'm studying the association between age and gray matter volume in adolescence, I just collect participants uniformly across the age range. Okay, but you can actually increase the variance of your covariate by sampling instead at the tails of the distribution. And that's because this effect is assumed to be linear, so you get more power by just increasing this standard deviation of x. So you can do this, and it doesn't really change the interpretation of the correlation coefficient at all, because your parameter is the same. So the relationship between age and the gray matter volume is the same here. You've just changed how you're sampling the data and increased your standardized effect size. In general, this relationship might not be linear. And then you might compute, for example, the correlation between your image outcome, y, and then some unknown function of your maybe high dimensional covariate x. And this correlation coefficient is equal to this once you do some, do the math and go through some simplification. So you can still see it's a variance of it's a ratio of the variance of your predictor, mu of x, so your linear predictor, and your the variance of your outcome. So mu of x is an unknown function that's just the expected value of y given the covariates. Okay, so what we wanted to do in this study is explore design features in brain-wide association studies that are associated with larger effect sizes. And we're going to do this empirically using data from some of my collaborators. Any questions or comments so far? Okay, so sorry, I'm dealing with a thing at home. I'm moving next week, so I've got all this stuff going on. So to explore effect sizes in this context, I'm going to talk about a robust effect size index. So this is the effect size we're going to use, and it has several advantages over other commonly used effect sizes. One, it's defined from M estimators, so it applies to many different model types. So you don't have a different effect size for a bunch of different models. Second thing, it's a robust effect size index. So robust in the sense of like sandwich covariance estimators, meaning that your effect size is consistent even if your model is misspecified. Three, it easily handles covariates and is computed from commonly reported test statistics. So it's easy to compute from published studies, and it's okay if you have covariates in your model, no problem. Finally, when the model is correctly specified, the robust effect size index, RESI, is functionally related to common effect sizes that you would use, such as Cohen's d. And this is something we developed for some other research we were doing, and that we proposed it in 2020, and then my student Kaidi Kang developed a confidence interval procedure, which I'll talk a little bit about because it's kind of cool. So to define the index, assume that beta is your parameter of interest, and that it's not equal to some null value, beta 0. Okay, then you can compute your walled test statistic like this. So T sub M1, this is your test statistic. M1 is the dimension of beta. So this is a chi-square statistic on M1 degrees of freedom. And then this test statistic is equal to the inner product between your estimated parameter minus beta naught, which I'll call the reference value. And then you have sort of your inverse variance of your square root n beta hat here. Okay, so under some assumptions, this test statistic is chi-square distributed on M1 degrees of freedom. So that's the numerator degrees of freedom. And then there's two things that control the non-centrality parameter of the chi-square statistic. One of them is the sample size, and then the other one is this thing here. Okay, so this is the distance between, or the squared distance between your true parameter and the reference value. And then it's standardized by the variance here. Okay, so this variance is the variance of root n times your estimator. Okay, so the important part is you have two components to this non-centrality parameter. One is n, one is this thing that doesn't depend on the sample size, it's a parameter. So we define the robust effect size as just that blue part. So the part of the non-centrality parameter that doesn't depend on the sample size. Okay, and so what I alluded to, the first component of that statistic is just measuring the distance between the truth and whatever your reference value is. And so this thing is dependent on what parameter you're what parameter you're estimating, but it's also dependent on what study population you're studying. And changing this parameter would actually change the interpretation of the effect size, right? So if like you add covariates to your model, this effect size will have a different interpretation than if you don't have covariates in the model. The second component of this thing is the variance thing in the middle. And so this is just standardizing beta so that it's unitless. But this component is dependent on many things. So it's dependent on the study population. For example, the variance, it's dependent on the second moments of your covariates and your outcome variable. And I'm going to argue that changing the variance of your covariate doesn't really change the interpretation of the statistic. It just changes basically the efficiency of how you're estimating beta. Okay, and it also incorporates estimation efficiency due to other things. So there's infinitely many estimators for beta hat, and so if you are using a more efficient estimator, this thing will get sigma beta will get smaller, so a sigma beta inverse will get larger, and that will make your effect size larger. Oh, so I wanted to just take an aside here. So this is the effect size index that we're going to use for the rest of the talk. I wanted to talk about how to construct confidence intervals for this effect size and other effect sizes, because it's kind of cool work that Kaidi did that sort of calls into question other approaches for constructing confidence intervals for effect sizes. So an estimator for the effect size, this is the squared effect size, can be obtained from the test statistic. So if T sub M1 is your chi-square test statistic, you can get an estimator for the effect size or the squared effect size using this formula. So it's a very easy formula. And then sort of intuition would suggest, because your T sub M1 is you might expect it to be asymptotically chi-squared distributed on M1 degrees of freedom with this non-centrality parameter, then you could, it might suggest that basically you can use a chi-square distribution to construct confidence intervals for the effect size, because assuming this asymptotic approximation. But it turns out that approximation is not correct due to the fact that you have to estimate the variance of beta. And I've got some pictures to try and show this off on the next slide. But I found this to be very non-intuitive. Basically, your chi-square approximation that you use to do hypothesis testing or construct confidence intervals for your parameters, that type of approximation won't work for effect sizes. Okay, so again, this is just the same sentence from the last slide. So intuition seems to suggest that your chi-square statistic under the alternative is chi-square with M1 degrees of freedom and N with non-centrality parameter N times the effect size. Okay, so here's what that approximation looks like under the null. So this is when things work okay. And so I have different sample sizes. Then the x-axis is the test statistic values, possible test statistic values, and then the y-axis is just the PDF of the test statistic. And I've given you several different test statistic distributions. So black is the chi-square distribution. The red dashed line is an F distribution. So this is on 1 degree of freedom, M1 equal to 1. So the F distribution is asymptotically equal to the chi-square distribution. And then the blue dashed line is an F statistic, but now the covariates are random in each across samples. So basically this means like you're conducting an observational study, and in each study, you're collecting data on X, and then you're also collecting data on Y, and they're both random. So what you see here basically is what we would expect based on the classical theory that we use, which is that your F statistic converges to the chi-square distribution as the sample size gets large, and then this F statistic, when the covariate is random, also converges to the same thing. So everything works out in this particular case when the effect size, the true effect size, is 0, meaning this is under the null. Okay, so what I've done here is I've changed the effect size to 0.25, which is a medium size effect size. And what happens is that your chi-square statistic, chi-square distribution is no longer appropriate to use in small samples or asymptotically. So here, just comparing the black line to the F statistic distribution, as you increase the sample size, then you see that these lines actually, the densities are actually not equal to each other, and they get further and further apart as the sample size gets larger. And they get further apart as if the effect size is larger too. So what this basically means is that you can never use a non-central chi-square distribution to construct confidence intervals for an F statistic where you don't know the variance, where you're estimating the variance. So no chi-square confidence interval will work for effect sizes. The other thing that sort of blew my mind is that, looking at the blue dash line, this is the distribution of an F statistic when your covariate distribution is random as well. And that even actually differs from the F distribution. So you can see that the blue has slightly heavier tails than the red. So that means that, again, you can't use the F distribution to construct confidence intervals for an F statistic based on an F statistic when your covariate is random. Okay, so never, no, in classical, the classical approach for constructing confidence intervals for non-effect size parameters, it doesn't matter matrix is random or not, but here it matters. So consequence of this is that chi-square F and T confidence intervals that are used for effect sizes fail. And that's shown here. So this is simulations that we ran in Kaidi's paper. And basically, if you look, the columns are different effect sizes. And then the rows, so the first row is the chi-square, using the chi-square distribution to construct confidence intervals. The second row is the F distribution, and then the third row is a bootstrap procedure that we proposed to construct confidence intervals. And then there's different types. We consider parametric test statistics and robust test statistics too. And basically what you see is that the chi-square distribution never works for when you're estimating the variance. So it always has below the nominal coverage. The F distribution doesn't work here either, because the covariate distribution is random. So, you still have below the nominal coverage. And then if you use the bootstrap procedure, which is just a non-parametric bootstrap, you get the nominal coverage that you need. So, so I thought this was cool because sort of the implication of this is that, all the software packages are using either this chi-square or this F distribution to construct confidence intervals for effect sizes. But they're doing the wrong thing. They're not going to have nominal coverage. So if you want to use correct confidence intervals, we wrote an R package for this. This was written by one of my students, Megan Jones, and it uses this robust effect size index, and uses bootstrapping to get valid confidence intervals. It's up on CRAN. We're working on a paper now. It's actually on archive if you want to check it out. And so it basically, you can take any model and then put it into this, the RESI function. I don't have an example that I showed here. And then it basically gives you model summary results as you would expect. So all of this stuff over here is what you normally get from your model summary. And then it just adds in the effect sizes and 95% confidence intervals. And this works for most model types that are available in R. Okay, so that's Megan Jones. She wrote that package. Okay, so that was sort of a detour just about constructing confidence intervals for this effect size index. So the RESI is what we're going to use to do to look at effect sizes in brain-wide association studies. We scaled the RESI so that it's proportional to 1/2 Cohen's d when the model assumptions are correctly specified. Cohen, in the 80s suggested different small, medium, or large effect size values. And then this is just the suggestions he recommended on the Cohen's d scale converted to the RESI scale here. Of course, these are rules of thumbs, a rule of thumb. because, what is a small, medium, or large effect size is going to depend on the field. And, what people are saying now is that effect sizes in brain-wide association studies are much smaller than we initially thought. So maybe in BWAS, we might consider 0.1 to be a medium effect size or something, but it's just a rule of thumb. Okay, I think that's the preliminary stuff I wanted to mention before we get into the data analysis.
Simon?
Yeah, go ahead.
So probably I missed, but are you suggesting like bootstrap as your like methods to construct confidence interval?
Yeah, it's a bootstrap.
And probably I also missed, but like where do you show your effect size that are like based on the robust or based on the parametric approaches? Can I?
Yeah, you mean for the coverage? Well, I have it here. So the I didn't go into detail, you didn't miss it. So there's an Oracle version that we showed here. This is if the variance is known, if you're treating the variance as known. The plus is a parametric estimator of the variance. And then the triangle is your sandwich robust covariance estimator for the variance. And then each row here is a different method of constructing a confidence interval for those different types of test statistics.
Okay, yeah.
Did that answer your question?
So the way you use the M estimation is to construct the sandwich covariance estimator for the like your robust index size, but what does it really like mean in context? For example, if you construct a like linear regression model, and we are basically assuming that this model for the linear regression is correct, right?
For the so in the parametric one, you're assuming that you homoscedasticity essentially is your assumption.
Yeah. So what could be the source of misspecifications in BWAS that you're considering? Basically, like in BWAS, I to me it's more like constructing like multiple linear regression compared to the like simple linear regression.
Oh yeah, so it's sort of in BWAS, I'm going to be thinking about I'm going to be sort of thinking about the univariate situation. Whether you choose to use a robust estimator of the variance or a parametric estimator of the variance doesn't really, like, it's not going to be so it will, like, the robust one is the only one that will be consistent if your model is incorrectly specified. But like the difference isn't really important for the context of the analyses I'm going to show you. I'm just telling you this is the effect size index I'm going to use to look at, to compare different types of designs for brain-wide association studies.
Okay. And can I ask like one more, like from like this figure?
Mm-hmm.
I mean, like from my very intuitive understanding of the like robustness, I expected the robustest version of the like the coverage of your like robust version like could be closer to like 0.95, especially like if you see the like last column, last row. But it seems like there's some kind of like undercoverage happening in your like robust version, right?
Yeah, this is a Bayesian bootstrap, and it doesn't seem to work quite as well as the boots. So we use this third row in our software, which is just a classical bootstrap. And we also propose this Bayesian approach just for people who want a Bayesian interpretation, but it appears to not do quite as well. So that could be part of our implementation, or there it could be some theoretical property about the Bayesian bootstrap. I'm not sure.
I see. I see. Yeah. Thank you.
Mhm. Thanks for the question. The data set I'm going to use today is called the Lifespan Brain Chart Consortium. This is an awesome data set by a colleague, Aaron Alexander Block, who's at CHOP. It includes over 100,000 human participants. Some of the data are longitudinal. Just a reminder, our goal here is to identify design features in brain-wide association studies that are associated with larger standardized effect sizes. So we're going to perform a meta-analysis of 63 different data sets that are included in the Lifespan Brain Chart Consortium, which includes a subset of those is 16 of those include longitudinal data. Okay, and so their paper was published last year in Nature, they were looking at normative growth curves using Gamel's models. And you can check out their paper, it's really cool. So what we wanted to do is perform a meta-analysis to look at design features associated with larger effect sizes. So for each of the 63 data sets in the study, what we're doing is fitting a linear model, or for longitudinal data, we're going to fit a GEE. And we're modeling the imaging variable, YI, as a nonlinear function of age plus sex. And Y was, we take it to be gray matter volume, subcortical gray matter volume, or white matter volume. So after we estimate these models, we can get the standardized effect sizes for each term in the model. The standardized effect size that we're using is the RESI. And then we fit a meta-analysis model such as this. So the effect size S, so this is the RESI for age, and maybe this is on gray matter volume for study D or data set D, is a function of the design. So this is an indicator variable that indicates whether it's a cross-sectional or longitudinal study. And then we are adding nonlinear terms that quantify that summarize characteristics of the population we're studying in each data set. So we model it as a function of the mean age of that data set, the standard deviation of age in that data set, and the skewness of age in that data set. Okay, so this is a meta-analytic model, and the outcome is the effect size for that study. We're also doing this is a weighted regression because the data sets are all different sample sizes. So we're weighting by the inverse of the variance of this effect size. Before we did any of this, so within the data sets, there were some of the data sets were collected from different sites, and we applied site correction using combat or long combat if it was a longitudinal study. So here's just showing some of the study level data. So you can see there's big variability in the population that was applied to. So ABCD is kids age there's been collected over 1 to 2 years or something. So it's a very small age range, whereas some of the other studies have a much larger age range they're looking at. And then, you can see males and females are colored differently. And then we're fitting, these are just low S smooths, but we're fitting a nonlinear model to each of these data sets. Okay, so we're estimating parameters from here and pulling the standardized effect sizes out and then fitting them in a meta-analysis that's shown here. So here's these are partial regression plots of the fitted values. So I'll start by looking at standard deviation of age, because just based on our knowledge of statistics, we know that the standard deviation of the covariate should be related to the effect size in some way. Okay, and so what we see, this is the effect size for the age gray matter volume association. So as you increase the standard deviation of age, you see a linear increase in the effect size. So basically this means a sample studies that have a wider age distribution have, you know, a systematically larger increase in the effect size. So 1 year increase in the standard deviation increases the RESI on average by 0.1. Okay, so and 0.1 was, I called it a small, that's a small effect size. Okay, so that's a really easy way that you could increase the power of your study by sampling wider distribution. Okay, we also looked at, let me see what longitudinal design. So does conducting a longitudinal study help when you're studying age? These results are suggesting it does. So longitudinal studies have a systematically larger effect size. You know, it's almost 190% increase in effect size. And that's a 0.26 larger effect size. So that's going from, you know, 0 to a medium effect size when you collect longitudinal data. You know, another thing that we thought was interesting here is that your effect size varies smoothly through development. One thing in particular that was cool is like during like late adolescence, early adulthood is when you have the largest effect size, and then also in later adulthood. So these are the times when like changes in age are most strongly related to changes in gray matter volume. And then each row here is looking at subcortical white matter as well. You know, gray matter, total gray matter, subcortical, and white matter. Okay, and we see a similar design effect across studies. Okay, so this indicates that two things affect effect sizes in brain-wide association studies here in this particular example. One is longitudinal designs. So longitudinal studies have systematically larger effect sizes. So one question is like, this is almost like a confounder in when you're doing a meta-analysis. So if you're combining effect sizes across studies, there will be this systematic differences. So one follow-up question we had was whether we can adjust for this in a single study. And then the second thing is, the second conclusion is that the standard deviation of the covariate is strongly positively associated with the outcome. So this should come as no surprise to you know, us as statisticians, because whenever we do a power analysis, we see how all these, you know, factors contribute to effect sizes and power. So a natural question is that if you, you know, this was just a meta-analysis, so sort of looking post hoc, natural question is, you know, can we sample in such a way to increase standardized effect sizes? So let me just look at this first question here first. So can we adjust for differences between longitudinal and cross-sectional studies in a single study? So this is what I said, longitudinal studies have systematically larger effect sizes. So what we introduced to compare cross-sectional and longitudinal effect sizes in a single study is something we call the CS-RESI, cross-sectional RESI, that you can estimate within a longitudinal study that lets you see what the effect size would be if you had conducted your study cross-sectionally. And so basically we have a theorem to derive this estimator and show that it's consistent. But all, this is your estimator of the effect size. Basically, all you need to do differently is change how you're estimating the covariance of the parameter. So you use the same parameter that you had for the longitudinal design, but now you adjust it to estimate the covariance as though you had collected the data cross-sectionally. So kind of a really cool feature about this is that in your single study, so you collect a longitudinal data set, you're reporting your results, you can actually see whether your study design gave you better, a larger effect size than what you would have had if you conducted your study cross-sectionally. And so that's what I'm showing here. So this is a forest plot of all the effect sizes across the 63 data sets. The top 16 are the 16 longitudinal studies shown in yellow. And then we've computed for those longitudinal studies, the yellow is the longitudinal RESI. So that's the robust effect size index accounting for, it's a robust effect size index conditional on your longitudinal design. And then the turquoise one is the robust effect size index adjusted for your longitudinal design. So that's your effect size if you had conducted the study cross-sectionally. So we can zoom in just to make it easier to read on the subset of studies that we conducted longitudinally. So this is, again, the effect size for gray matter volume age association. And you can see that, like, in general, conducting a longitudinal study gives you, for age, gives you a larger effect size on average. It's not always true. There's some studies that, where there's sort of a loss of efficiency of conducting the longitudinal study. We're looking into these. Like, I think there's some artifacts in this data set that sort of made, like, make the longitudinal design less efficient than the cross-sectional design. In general, it's better to conduct a longitudinal study of age. Okay, so we can look at the analysis table. This is the sort of the summary table for the meta-analysis. And so if you look at the top, this is conducting on the yellow ones. So that are, these yellow ones are known to be affected by whatever the longitudinal design is. And this is, this test is testing what I showed on the last slide, essentially, that there's a significantly larger effect size in longitudinal studies. So it's 0.26 larger, that's what I showed. And then if you adjust using the cross-sectional version of the RESI, now there's no longer a difference between cross-sectional and longitudinal studies. Okay, so that sort of, there's two purposes for this. One, it's going to make your meta-analysis easier because now you have an effect size that's comparable across longitudinal and cross-sectional studies. Two, it's going to, in an individual study, allow you to quantify how much benefit you got from conducting the study the way you did. So we can also look at this for the other variable in the model, which was sex. So sex, for the most part, doesn't vary across the lifespan. So collecting multiple measurements on one subject will have, that covariate will have a low variance. And so you wouldn't expect to see too much benefit in looking at longitudinal studies for age. And that's pretty much what you see here. So again, like, for the most part, all of these effect sizes are pretty consistent across the longitudinal and cross-sectional studies. So there's not much benefit of studying sex in a longitudinal study, which, should make sense. Okay, so this was looking at, again, a meta-analysis across studies. So to answer that second question, whether we can modify the design of a study to increase effect sizes, we basically use bootstrapping to do this. So this is a study of the UK Biobank data. And what we did is we sampled from that data set to increase the standard deviation in the bootstrap samples in order to see whether that leads to an increase in standardized effect sizes. And so the way we did that is using sort of this these different density functions for sampling. So basically we selected people from the tails of the distribution with varying probability. So the first one is uniform sampling. So that's as the study was conducted. The green is linearly sampling the tails of the age distribution more heavily. And then the blue is quadratically sampling the tails of the distribution more heavily. And so sampling in this way naturally increases the standard deviation of age. And then you also see that there's, as expected, an increase, a corresponding increase in the effect size. So this is just to say sort of what we know from formulas, that increasing the sampling variance of the covariate will give you a larger standardized effect sizes. So a natural question we had after this is, what about longitudinal studies? So what types of longitudinal studies are going to be beneficial for this particular variable, which is age? So to do this, we can actually apply these same 3 sampling schemes, so weighting the tails more heavily between subjects and also within subjects in the we did this in the Alzheimer's disease neuroimaging initiative data. Okay, so now there's sort of colors here that represent the same thing. So red is uniform sampling, green is sampling linearly the tails more heavily. The blue is quadratically sampling the tails more heavily. So those are shown in 3 different colors. And then we did the same 3 schemes sampling the within subject measurements from the ADNI here. So scheme 0 is sampling uniformly from measurements within the subject. Scheme 1 is sampling linearly the tails within subject more heavily, and scheme 2 is sampling the quadratically within subject more heavily. Okay, so we have the same design we're doing within and between. And then we can look at the cross-sectional resi. So this is the effect size if you had conducted this study cross-sectionally, and longitudinal resi. This is the effect size conditional on the design of the study, the longitudinal design of the study. So basically what we see, this is to be expected. So if you look at the cross-sectional resi, changing the within subject design doesn't change anything. So as you go across these panels, the lines are all pretty much the same. And that's because this is adjusting for whatever your within subject design is. Again, you still see that when you increase the variance across subjects, you get a corresponding increase in the effect size. What we were surprised to see is that for the longitudinal resi, sort of increasing within subject variance didn't really increase effect sizes either. So this was a little bit surprising to us, that the within subject sampling standard deviation had minimal effect, just based on formulas that you can get from your longitudinal textbook. You would expect increasing the variance within subject to increase effect sizes. And part of why we think this isn't happening in this data set is that the within subject variance is relatively small. So between subject variance in gray matter, in age accounted for 77%, 81%, and 84% of the total variance in the different between subject sampling schemes. So that means that the within subject variance only accounts for less than 25% of the variance. So adjusting how big that is, it's still relatively small. It's going to have a relatively small effect on effect sizes and thus power and replicability. Okay, so that's looking at between and within subject standard deviation on effect sizes. Well, so then after that, we thought, well, let's look at what if we simplify the design a little bit. So instead of, sampling all the time points that they had in the ADNI, which I think was an average of, I don't remember how many, it was more than 2. So there were several time points per subject. So what if instead, without changing the covariate standard deviation, we sample 2 time points for each subject, instead of sampling, all the time points for every subject. Okay, so what I'm showing on the left is what I had on the last slide. And what I'm showing on the right is the same thing, but now just looking at this ADNI again, but only sampling 2 observations. And what we see again is the cross-sectional study sort of has a minimal change. But then in the longitudinal study, we basically see using 2 observations, you get almost as much benefit using only 2 observations as you get collecting multiple measurements per subject. So this sort of suggests, that, well, maybe I have it on the next slide. So sort of our conclusions here are that sampling more heavily at the tails of the target distribution across subjects increases standardized effect sizes, and this will increase power and also increase replicability. And then the second take-home point was that basically collecting a single additional longitudinal time point, so collecting 2 per subject instead of 1 or more, confers as much benefit as collecting multiple measurements per subject, conditioning on that your within subject standard deviation remains the same. So this was looking at the age covariates. I'm aware of time. I'm gonna, I've got 3 minutes left, so I might skip some stuff. But so the next thing we had done in this study is to look at other types of covariates. So brain-wide association studies aren't just about studying age, they're about studying psychometric measurements and how they associate with the brain. So that's what we looked at next. So to do that, we looked at the ABCD study, where we had a whole bunch of psychometric measurements. So we have still, again, our 3 main cortical volumes that we looked at. And then we had 10 psychometric variables from the ABC data set. I'm just showing 3 here. So the first is crystallized composite intelligence. Then there was a flanker task, and then oral reading recognition task. And then we're showing I'm forgetting what the colors are. Oh, so these are 2 sampling schemes again. So one is the turquoise one is increasing the between subject standard deviation. And then again, the different panels are showing what happens if you increase the within subject sampling variance. So the right column is the within subject sampling variance. And then, sort of the left 2 panels are the cross-sectional effect size. We wouldn't expect within subject sampling to affect that value. And then on the right is the longitudinal resi. So one really surprising thing that we saw here is that between subject standard deviation still improved effect sizes. So that wasn't surprising to us. So, you're comparing within each panel, the red to the blue. However, when you look at changing the within subject variance, it actually reduces standardized effect sizes. So they're pretty consistent here, but you can see, like, if you look at the longitudinal resi and compare that to the cross-sectional resi, you actually see a detriment of collecting longitudinal data here. So the effect size actually goes down for crystallized composite intelligence. So that's a little bit concerning that, doing a longitudinal analysis actually hurts you in this case. So what's the deal with that? So part of it, we believe, is related to, so we know that longitudinal analysis, there's sort of 2 types of effects that can exist. There's a between subject fixed effect, and there could be a within subject fixed effect as well. And basically, if those are different, you can go through the math and see that if those are different, it can actually be conducting a longitudinal design could be harmful to you if you're modeling them with only one parameter. We know it's not related to efficiency because basically with very minimal assumptions, conducting a longitudinal design always increases efficiency. So we know it has something to do, basically the change has something to do with conducting with the effect of within and between subject relationship between the outcome variable and the covariate. Here's what that looks like in the ABCD data. So this is the x-axis is the change in the covariate measurement for these are 6 psychometric variables. And then the y-axis is the change in gray matter volume. So you can see the changes in the psychometric measurement are pretty much independent of the changes in gray matter volume. And so that's why you're not seeing a benefit to conducting a longitudinal study here. To contrast that with a change in age versus change in gray matter volume in ADNI. So here, this is showing that within subject changes in age are associated with within subject changes in gray matter volume. So this means that, collecting longitudinal data in this case is going to be helpful to you. Collecting longitudinal data here will improve efficiency, but because the within subject effect is different than the between subject effect, then it actually hurts you in terms of estimating the parameter beta. So all of this was just to say that, we sort of think of standardized effect sizes as parameters, but they're actually modifiable by study design. In particular, changing their sample population variance can increase effect sizes, which in turn would increase power and replicability. Most of your benefit to a longitudinal study seems to come in the second measurement, at least for the things that we looked at. And then obtaining covariates that are, the values are farther apart is gonna be better. Obviously, sometimes we can't know. If you have a psychometric variable, you don't know what the next measurement is gonna be for that subject. And then you have limitations like, if you're sampling subjects with high intra-subject variance in a psychometric variable, that could be indicative of psychopathology, which is probably not, you don't want to select people with high variance in that case. So, and then, there's going to be other features, like if your outcome is gray matter volume, which you expect to change slowly over time, having short-term large changes in psychometric variables are not likely to be correlated with that thing that changes slow over time. So that's sort of what we've been working on. And by we, I mean Kaidi, who's shown here. Kaidi's in his 4th year. He does amazing work, and he'll be on the job market, I guess, after next year. Megan wrote the Resi package. And I particularly want to thank Aaron and Jacob, who organized the LBCC data set, and my colleagues here, Jonathan and Rand, who worked on this sort of the statistical intelligence part of it with me. And then this is the funding source for this work. So thanks very much. Sorry for going 5 minutes over.